Hi everyone, thanks for the opportunity to share my work with you. My name is Meiqi Niu, and today I'm going to talk about the cyto and receptor architectonic organization of macaque primary and secondary somatosensory cortex. The somatosensory cortex is involved in the processing of sensory information from across the body, such as touch, temperature, and pain. The monkey primary somatosensory cortex is located on the posterior wall of the central sulcus and encroaches into the free surface of the parietal lobe. Over the last few decades, researchers on monkey has defined four strip-like areas in this region, namely areas 3A, 3B, 1, and 2. Each of these areas contains a separate body surface representation, which is arranged from the foot in the medial cortex to the face and tongue on the lateral cortex. The secondary somatosensory cortex is found on the lateral parietal and is almost completely hidden in lateral fascia. The monkey secondary somatosensory cortex consists of at least two areas S2 and parietal ventral area PV. An understanding of how the somatosensory cortex processes sensory information depends in large part on knowing how it is organized. A complete and detailed parcellation of the corresponding region can offer improved understanding of the biological substrate of brain function, dysfunctions, and behavior. Therefore, the aim of the present study is to provide a refined architectonic map of macaque primary and secondary somatosensory cortices. We not only wanted to analyze the cytoarchitecture of this brain region, but also to obtain some information which could help us understand the combined structure and function organization of this region. Thus, the idea came up to use neurotransmitter receptors. Receptors are key molecular which enable signal transmission between nerve cells and are heterogeneously distributed throughout the brain. Importantly, the function of cortical area depends on a well-toned balance between different receptor types. Thus, by visualizing multiple receptor distribution pattern, we can not only identify the location and the extent of the brain regions, but also get a glimpse of their functionally relevant organization by assessing the degree of the similarity of the multiple receptor balance in each identified area. So let's take a look at the method we used. We analyzed the brains of 3 adult macaque fascicular wrist monkeys. Brains were removed during autopsy. Each hemisphere was deep frozen and sectioned into corona sections of 20 micrometer thickness. Alternating sections were incubated with tritiated ligands or stained for cell bodies. As an example, here I show a group of sections from the same rostro-caudal level. For this group of sections, we can obtain information of 15 different receptor types and the corresponding cyto and myelin architecture. That means, in this very tiny brain region, we have more than 15 modalities to study. After digitization and color coding, we obtain a clear visual impression of the regional and laminar distribution of multiple receptors, which were used to identify the borders between different areas. An observer-independent and statistically testable method was applied for detection of the potential area borders in both cyto and receptor architecture. The potential border was accepted only after confirmation of its statistical significance. Moreover, the biological relevance of the potential border was confirmed by comparing spatially corresponding borders from adjoining sections. For each identified area, we extracted the densities of all analyzed receptors for further statistical analysis. In the present study, 13 architectonically distinct areas were identified. For better understanding of the topographical relationship towards neighboring areas, here I show a series of corona sections superimposed with the corresponding areas. Within the primary somatosensory cortex, we confirmed the existence of previously described areas 3A, 3B, 1, and 2. Additionally, we identified medial and lateral subdivisions of 3A and 3B respectively. Furthermore, lateral part of 3B could be further subdivided into internal and external components, whereby 3BLI occupied the entire dorsal-ventral extent of the central sulcus, but 3BLE was only found in its dorsal portion. Within the secondary somatosensory cortex, 6 identified areas are arranged in 3 rostro-caudal zones. The caudalmost zone with the medial and lateral subdivisions of area S2, a middle zone with area PVM medially and PVL laterally, and the rostermost zone with area PRM and PRL. Here I show examples of the receptor architectonically defined borders of area 3BL. This figure depicts the distribution of GABA A receptors and corona sections through a macaque central sulcus. Blue code for low and red for high receptor densities. The borders between internal and external subdivisions of 3BL are reviewed by changes in GABA A receptor densities. All other examined receptors also review this border, either by overall density differences or by changes in specific layers. It's worth noting that not all borders are equally clearly definable by all receptor types. The absolute mean areal densities of all receptor types was then quantified and projected into the corresponding areas. That means, in addition to redefine a detailed architectonic parcellation scheme, in the present study, we further provide 15 receptor distribution maps of the macaque primary and secondary somatosensory cortex. One of the purposes of this study was to analyze the organization of the macaque somatosensory cortex, with a focus on the relationship between its molecular and functional segregation. So, for each of the identified areas, the densities of the 15 receptor types were visualized as receptor fingerprints. Each area has its own characteristic receptor expression pattern, and the similarities of their receptor fingerprints enable the segregation of these areas into 3 functionally relevant groups. First, multivariate analysis reviewed a clear segregation of primary and secondary somatosensory areas. Moreover, within primary somatosensory cluster, area 3AL and all subdivisions of 3B are found in one cluster, and 3AM, 1 and 2 in the other one. We have termed the former cluster as S1 proper cluster, since areas 3B, 1 and 2 form a hierarchical sequence of processing. 3B is thought to constitute the true primary somatosensory cortex, which has chemoarchitectonic features that are characteristic of primary sensory areas, while areas 1 and 2 had less dominant sensory features, which resemble those of secondary sensory areas. This notion is further supported by the segregation of all 3B subdivisions from areas 1 and 2. Now, I want to summarize my talk. First, we reevaluated the parcellation scheme of the macaque monkey primary and secondary somatosensory cortex, and created a 3D map which has been brought into stereotaxic space. Second, we created receptor distribution maps of this region for all analyzed receptor types. All those maps will be publicly available via the Human Brain Project and the BASA repository afterwards. At the end, we provided insights supporting a new functionally relevant organization model of this region. Last but not least, I would like to thank my colleague for their support, especially Nicola Palomero-Gallagher, and I want to thank you for all your attention. If you are interested to hear more about this talk, I'll be very happy to welcome you at my poster. Thank you very much.